This lecture will cover the classification of microbes, which will include both the taxonomy and phylogeny of microorganisms, which, as you can see from the picture here, will include lots of different organisms from many of the well-known kingdoms that you learn about in general biology. It turns out that taxonomy, which is the science of naming organisms, has been around for quite a bit of time. In fact, Aristotle is the first person to have tried to create a taxonomic system. In the 4th century BC, he published something called Historia Animalium, in which he categorized animals. So he didn't take on all of the organisms on the planet, but he took on animals. And in addition to categorizing them into genus and species names, he also differentiated them into these groups based on some unique trait that he perceived in them. So as you can see, he took animals and he divided them into two categories: the animals which had red blood and those without red blood, and then within those vertebrates, as we call them today, he categorized them into groups based on whether or not they had live young or viviparous, or if they had eggs, if they were oviparous, whales, fishes, birds, and so on and so forth. So though he didn't create the taxonomic scheme that we're familiar with today, he did come up with this idea of a binomial nomenclature, a two-name naming system, which we talked about in the first lecture, which has a genus name and a species name. And that idea was going to be picked back up on, but not actually for a very long time. In fact, it took until the 1700s for that idea of binomial nomenclature to really catch traction and be used in a larger system. And in 1735, this idea of binomial nomenclature was published in a book called Systema Naturae, written by Carolus Linnaeus, which employed that binomial nomenclature. But in addition to that, it divided all of the known organisms into three kingdoms, and Linnaeus made those three kingdoms the animal kingdom, the plant kingdom, and the mineral kingdom. Now, of course, red flag there, right? Minerals are not organisms, so that actually got thrown out. But animal kingdom and plant kingdom we have thanks to Linnaeus. And what we also have to thank Linnaeus for is this systematic categorization of organisms into kingdoms, phyla, classes, orders, family, genera, and species that we use today, where kingdoms are these very inclusive groups that contain lots and lots of organisms that have very little in common, and then all the way down to species, which are a group of organisms which share almost all of their characteristics. And for most of the organic world, the definition of species has been organisms that can interbreed and produce fertile offspring, which of course doesn't work for microorganisms all the time, because it turns out that many microorganisms do not engage in sexual reproduction, and so they don't fit that definition at all. However, the idea of species being a group of organisms that can interbreed and produce fertile offspring really means that you have organisms whose DNA is so compatible that it can come from male and female gametes, fuse into a fertilized egg, and then create viable offspring, which itself can also make viable female or male gametes. So it really does rely on genetic similarity. And so today what we do is we organize microorganisms into species based on their genetic similarity. Now, it wasn't long before the publication of Systema Naturae in 1735 got other people thinking about the world around them and how to categorize that world. So you can see here that in 1758, this tree of life produced by Carolus Linnaeus has lost that third branch, the mineral branch, and so he was categorizing organisms into just animals or plants. And about a little bit over 100 years later, Ernst Haeckel wrote The General Morphology of Organisms, and in it he proposed 4 kingdoms. He actually doubled the amount of kingdoms, and he did that because of all of the advances that had been made in microscopy and studying microscopic organisms. And what he found was there were these groups of single-celled organisms who were plant-like and animal-like and sometimes fungi-like, although at the time there was no group called fungi. And they had nuclei, and they were oftentimes unicellular. So he created this group called the protists. And then he also created this group called Monera, which included everything he found that didn't have a nucleus. And I think one of the most amazing things about Ernst Haeckel is the phenomenal drawings that he produced when he created this system and published his works. And if you have time, and if you're at all interested in artwork, you've got to check out his Kunstformen der Natur. Just Google that and see some of the amazing drawings that he put together in order to make the case for this system of classification. All right, and in keeping with that 100-year span, we go fast forward again to the 20th century. In 1969, Robert Whittaker proposes adding a 5th kingdom, fungi, to the tree of life, which is kind of interesting. You might wonder, what were people, where were the fungi? They were actually grouped with the plants, probably because a lot of them are green, but strangely enough, none of them photosynthesize. All fungi are heterotrophs. They eat food just like animals do. So kind of a weird thing to throw them in with the plants. I think it was just a matter of not really knowing what else to do with them, but that was solved in 1969 when Robert Whittaker added that 5th kingdom. And what Robert Whittaker also did was look at these organisms and say, there's just such a huge difference between kingdom Monera, which is comprised of prokaryotes, and all of the rest of these kingdoms, that really it needed a different and bigger classification distinction. And the classification distinction that he came up with was something that was larger, more inclusive, above the level of kingdom, and he called them empires. And he divided all of life into empire prokaryota, which at that point in 1969 was just kingdom Monera. Today, we know that that includes 2 groups of prokaryotes, bacteria and archaea, but prokaryota was an empire built just of kingdom Monera, whereas empire eukaryota was an empire that was made of kingdom Plantae, kingdom Protista, kingdom Fungi, and kingdom Animalia. So he added this idea of empire, which is going to play heavily into modern taxonomy, because he's not the only person that thought some other distinction needed to be made to differentiate just how different eukaryotes and prokaryotes are from one another. However, most of that ability to understand the differences between prokaryotes was not going to happen until the genetic revolution, where people learned about DNA, understood how to work with DNA, how to find it, how to sequence it, how to understand it. And so it really wasn't until the 1970s that people started to take a serious look at the taxonomy of prokaryotes, because it was sort of thought up until that point that all that could be known was known, and what could be known was not that much. There was the shape to consider, right? Did you have coccus or bacillus or a spirillum or a vibrio or a cocobacillus or a spirochete? Did that organism stain gram-positive or gram-negative, or was it an acid-fast bacterium? Did it have endospores? Did it have a capsule? All of those things were very superficial. They were just based on a couple of characteristics of the cells. And it turns out especially gram staining and morphology and endospore staining, actually all of those things are found all throughout kingdom Monera. So to say that 2 organisms are both gram-positive is really not to say that there's all that much about them that's similar. The other thing that was used traditionally to name and categorize microorganisms was their physiology. So what did they eat? What were they able to eat? What are they not able to eat? Do they photosynthesize? Are they detritivores? Do they live in the human gut? What do they do? What do they eat? How do they make their ATP? And today, though, the genetic revolution has allowed microbiologists to understand much more fully just how these organisms should be classified based on genetic similarity. And in 1977, a pivotal experiment was done by Carl Woese and George Fox, where they started looking at the ribosomal RNA component of ribosomes. And this was a fantastic thing to use as a comparison because all organisms have ribosomes. Now, they may not have the same ribosomes, but they all have ribosomes. All of their ribosomes are made of a small subunit and a large subunit. So Woese and Fox decided that they would get their hands on the RNA from as many organisms, small subunit, as they possibly could, and they started sequencing this ribosomal RNA, and when they did that, they were able to create one of these phylogenetic trees, these trees based on organism similarity, not just of animals or plants or bacteria, but a tree that encompassed all of the known extant organisms on the planet, all of the living organisms on the planet. And when Woese and Fox completed their work and they had put together this phylogenetic tree of all of the extant organisms on the planet, they found something really remarkable. And that remarkable thing that they discovered was that these seemingly very similar prokaryotes, all of which have no nucleus, some of which stained gram-negative, some of which stained gram-positive, were in fact really two distinct groups of organisms, that themselves were so very different that Woese proposed that there shouldn't just be two classifications above kingdom, there should be three, and he named those three domains. So you can think about the domain as the modern equivalent of Whitaker's empires, and he said it's not just that there's empire eukaryota, there's domain eukarya, which includes all of the eukaryotes. But if we group them together, and each of these lines indicates the amount of difference between two organisms, the eukarya are all together, and in fact, all of the multicellular life forms that are known on the planet today are in the teeny tiny tip of domain eukarya. Most of domain eukarya, most of the diversity there exists among protista, which is really remarkable. And that doesn't mean that you and a fungus and a slime mold are the same. You're very different, right? And you can use that difference as a context point for the fact that if you have such a short line between you and a slime mold, imagine the difference between all of these archaeans, shown here in red, and all of the bacteria shown in purple that were previously grouped together in one kingdom Monera. So Woese proposed not empires, but domains of life. And he said we need three domains. If we're really going to do justice to the amount of difference that exists between all of these prokaryotes, we've got to have three domains. We have to have domain bacteria. We're going to take some of these archaebacteria that we had grouped into this area before and say, no, they're so different that they get their own domain. They are domain archaea. And then we're going to take the four eukaryotic kingdoms and place them into domain eukarya. So this three-domain system, which was proposed in 1977, has now supplanted the Linnaean system of taxonomy, which starts with kingdom and gets more exclusive from there. Now we have domains, and then kingdoms, and then phyla, and then classes, and then families, and then genera, and then species. So kingdom monera is kind of an antiquated word now. In fact, sometimes you'll hear people say kingdom eubacteria and kingdom archaebacteria, just to give a nod to the fact that you can't have two organisms in two different domains who then somehow go into the same kingdom. So let's take a look for just a moment at these three domains and what makes these archaeans so different enough that they deserve their own domain. Let's first compare the archaea and bacteria to the eukarya. So if you look at the chromosome, archaea and bacteria have typically a single circular chromosome and contain plasmids, whereas domain archaea have multiple linear chromosomes. Archaea and bacteria have what we call inclusions. They have no nucleus, they have no membrane-bound organelles, whereas domain eukarya has membrane-bound organelles, including the nucleus. Archaea and bacteria have highly variable modes of metabolism, meaning we have autotrophs, chemotrophs, chemoheterotrophs, all these words that we're going to learn more about later on that basically mean these guys are capable of eating all sorts of bizarre things, and they are the only organisms on the planet that are known to fix nitrogen, whereas domain eukarya tends to have those typical metabolisms that you learn about in general biology, that photosynthesis that's typical of plants, the respiration and fermentation that's typical of animals, and then reproduction is another area where archaea and bacteria are very similar. They both only undergo sexual reproduction, and the only exchange of genetic information that happens amongst them is something called horizontal gene transfer that we're going to be talking about in our next unit, whereas the eukaryotes of domain eukarya typically have both sexual and asexual reproduction. So at this point you might be thinking, well, these guys look like they're very, very similar, so why would we move them into two domains? Hold on for just a moment and let's look at what's similar between domains archaea and eukarya. All right, so archaea and eukarya, when we compare them, if we look at different things, more metabolic than structural, we find that the gene expression, the way that archaeans and eukaryotes undergo transcription and translation is very similar to one another, while bacteria undergo a totally different kind of gene expression. In fact, the start codon for both archaea and eukarya, you might remember that from general biology, is methionine, whereas for bacteria, it's formylmethionine. So it's a totally different amino acid that's used to start the translation of bacterial proteins. In addition to that, kind of an interesting thing, archaea and eukarya are both sensitive to diphtheria toxin, which is it's a kind of toxin called an AB toxin that we're going to be talking a lot about later on. Its name might sound familiar to you because it's the toxin that is produced by virulent strains of Corynebacterium diphtheriae, which causes diphtheria, and bacteria are all completely resistant to that toxin, while members of archaea and eukarya are both sensitive to that toxin and their cells are killed when exposed to that toxin. So you can see there's this conflict. There's a lot about archaea that's similar to bacteria, there's a lot about archaea that's similar to eukarya, and then there's a lot about archaea that is just archaea alone. And one of the really fundamental things about archaeans, which is not shared with anybody else, is that their cellular membrane is completely differently put together than anybody else on the planet. We're going to look at this in further detail in other lessons. Remember, we talked about this when we looked at the prokaryotic cell. Archaeans have ether-linked lipids in their phospholipid membranes, and bacteria and eukarya have ester-linked lipids. They all have unique RNA polymerases and they all have unique cell walls. In archaea, the cell wall is typically pseudopeptidoglycan if it's there. In bacteria, it's typically peptidoglycan if it's there. And in eukarya, we have all sorts of different things being used to make the cell wall. So lots of stuff that's similar between archaea and bacteria, lots of stuff between archaea and eukaryotes, and then finally lots of stuff that's archaean alone. So hopefully that gives you a little better idea of why in 1977 Woese and Fox proposed this 3-domain system to sit atop the kingdom system that we learn about in general biology. So as I mentioned, that new system of classification of putting organisms into groups based on similarity was based on sequencing of the ribosomal RNA, which is a nucleic acid, in any organism that they could get their hands on. And so that then laid the groundwork for using the sequencing of nucleic acids in all sorts of respects in order to classify not just microorganisms, but all organisms. However, it works really nicely with microorganisms because there's so little that you can choose as a characteristic to decide whether or not two organisms are similar to one another. So what you're looking at here is the nucleotide sequence, the RNA sequence for 4 organisms. Organism A, whose 12 base sequence reads CGUAGACCUGAC. Organism B under there in blue, organism C in yellow, and organism D in green. When you compare organisms A and B, you notice that out of 12 total nucleotides, you have 3 differences. And if you take 3 and divide it by 12, you end up with a difference between those two of 0.25. So if you look down at the bottom here, you'll notice that the sequence dissimilarity between organism A and B is noted as 0.25. Similar comparisons between A and C yield a sequence dissimilarity of 0.33. Comparison between A and D yield a sequence dissimilarity of 0.42. And so on and so forth until all 4 of those organisms have been compared to one another. And now you have an idea of how dissimilar they are. But what you really want to do is try to figure out not only how dissimilar is A to B, but how dissimilar is A to B, C, and D in one place. That's how you produce one of these phylogenetic trees. This is kind of a complicated process. Thankfully, in the modern age, we have computers that will take those sequence dissimilarities and build a phylogenetic tree out of them, where you can very easily and quickly see the sequence dissimilarities between each of these organisms. It's easy to see from this diagram that organisms A and D are the most dissimilar to one another, which was easy to see from the number as well. What's not so easy to see is how each of the organisms relate to one another when you have the 0.33 in 3 different relationships and the 0.25 in 2 different relationships. So what you can get from this phylogenetic tree then, again, like we did with Woese's tree of the 3 domains of life, is lengths on these tree branches that are equivalent to the amount of difference between the organism. Now, it's all well and good to construct a phylogenetic tree on your own with pen and paper when you're comparing 12 nucleotides between 4 organisms, but that was a much, much smaller example than what Carl Woese had taken on. What Carl Woese did was sequence about a 1,600 base pair sequence from every organism that he could get his hands on. And this amounted to thousands and thousands of organisms. So today, that 16S ribosomal RNA sequence has become the landmark sequence to compare any unknown bacterium or organism to a set of knowns in order to figure out where they lie in that classification scheme. And when you do that, thankfully you have a lot of technology involved in the process. Even the sequencing of the DNA can be done very cheaply by sending a sample of your bacteria to a lab who will then sequence the DNA and send it to you. And so what I thought might be an interesting thing to see is some of these actual data at work. So if you look at this figure, you'll notice along the top it says MLS1, and it has a series of nucleotides, T's and C's and G's and all that fun stuff. And then that continues at the top of that second block, and the third block, and the fourth block. So notice here we're looking at about a 500 base pair chunk of DNA, and the computer has been given the DNA sequence of this MLS1 strain. This is a strain of bacteria that I isolated from Mono Lake when I was doing my graduate work. And the computer is comparing that sequence to the sequences of all of these known organisms. And what it does, once it goes into its database and makes all of these comparisons, is it spits out this chart for you, which will give you the DNA sequence of your unknown and the DNA sequences of the 15 most similar organisms that it has in its database. And everywhere your organism and the other organisms is the same, you have this blue letter, and everywhere there's a difference, you have a pink letter. So you can see lots of blue, lots of similarity between these, but in that pink you see some differences. But the problem is when you're looking at 500 base pairs and 15 organisms, it's a heck of a lot harder to figure out who's similar to whom. Thankfully, technology comes to the rescue again. You hit a button and you ask the computer, and the computer makes for you a phylogenetic tree, which again shows you all of these similarities in pictorial form. So you can very quickly see that this strain MLS1 is very similar to this Borgoria red and Haloalkalophylla and Vacuolata and E. marina that all belong, not surprisingly, to the same genus, Ectothiorhodospira, that this organism was placed into. So this scheme that was pioneered by Woese and Fox has now become the standard for how to classify microorganisms into groups, and new microorganisms, new prokaryotes are being discovered all the time and are being subjected to this testing so that they can be placed into this scheme. Are they domain archaea? Are they domain bacteria? Are they domain eukarya? Which kingdom, phylum, class, order, family, genus, species do they belong to, right? So this nucleic acid sequencing allows us to cut out all the middlemen, not what's the DNA that makes you have the shape, or the DNA that makes you have the Gram-Stain reaction, or the DNA that makes you make a nucleus or make flagella or form endospores or mycolic acid or any of those things. It just looks straight at the DNA itself, and it uses that information and applies it to that same old classification scheme that Linnaeus proposed in the 1700s and you learned about in general biology. It turns out that modern microbial taxonomy does the same thing that we've always tried to do: split the organism into groups based on similarity of features or characteristics. We begin with all organisms and we quickly move them into one of three domains, which means these three domains, eukarya, archaea, bacteria, have very little in common. When you move to the level of kingdom, things get more exclusive. So some get ruled out, right? Amongst all of the eukaryotes, only those organisms which have a certain set of characteristics are members of kingdom fungi. And amongst all of the fungi, only those members who have characteristics in common can be members of phylum Ascomycota, and so on and so forth. So the phylum is more exclusive than the kingdom, and the class is more exclusive than the phylum, and the order is more exclusive than the class, and the family is more exclusive than the order, and the genus is more exclusive, and then finally you get to this group of organisms, Saccharomyces cerevisiae as an example, which are so very similar to one another that they've been grouped as a single species. And that happens throughout the entire known living world. That happens with fungi, it happens with archaea, with our Methanothermococcus okinawensis, It happens in bacteria with Escherichia coli. It happens within kingdom Plantae, kingdom Protista, kingdom Animalia. It's all the same, and it's all based today on similarity of DNA or other features. Now, having said that, what we need to realize is that this organism here, Escherichia coli, the genus Escherichia and the species coli, is something that was named and known long before these genetic analyses were known and used. And what that means is there are many, 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 many different strains of E. coli within this same species. So just like dogs have breeds, bacteria have strains. And if we take E. coli as an example, these are all the different strains of E. coli that have been found that are genetically distinct enough or morphologically or phenotypically distinct enough that they become separate strains. And kind of strangely, right packed in the middle of them are a bunch of Shigella species that apparently are very, very similar and in fact more closely aligned with Escherichia than with the genus Shigella that they were actually placed in when they were identified. So it's kind of thrown taxonomy on its head to have these new genetic ways of categorizing organisms and placing them into groups, because it's changing some of those historical phylogenetic groups. Thankfully, though, we do have a place where known prokaryotes have been cataloged for about 100 years, and that is David Berger's manuals, Berge's manuals of systematic bacteriology and determinative bacteriology. So Berge's manual of systematic bacteriology is a huge multi-volume set where Berger set out to just write and categorize every single organism that he could find. Berge's Manual of Determinative Bacteriology is a single volume work that's used to determine the identification of an unknown bacterium. And though we're not going to work with Berge's Manual of Systematic Bacteriology in this class, we are going to be using Berge's Manual of Determinative Bacteriology when we work with unknowns. We will be looking into this determinative bacteriology to ask what are the morphological and physiological characteristics that I can look for in an unknown in order to figure out who that unknown is. And that's going to be a really big tool for you later on in the semester. Now, that doesn't mean go out and buy that book. It's expensive and you don't need it. I will give you all of the pieces of that book that you need in order to get the class done. But I just wanted to show that to you and let you know that they have been cataloging prokaryotes, but a lot of that cataloging, as you can see, because it's older, is based on morphology and physiology. But nevertheless, the species are still grouped based on similarities. They might contradict the similarities that are found based on DNA, like we saw Shigella becoming part of E. coli genetically. However, when you look in the determinative bacteriology manual, Shigella is absolutely distinct from E. coli, and there are morphological and physiological ways of distinguishing them very easily from one another. One of the ways that the prokaryotes are cataloged has to do with the DNA in the only way that was available to people in the 1920s, and that is to have classified the organisms in the determinative bacteriology, which, by the way, will include both bacteria and archaea, because in the 1920s, archaea were part of bacteria. Remember, it wasn't until the late 1960s that Whitaker suggested this very distinct group of organisms that we call prokaryotes, and it wasn't until the 1970s that domain bacteria and archaea were established and accepted within the scientific community. So what they had available to them at the time was an ability to determine how much of the chromosome of a given prokaryote existed as guanine cytosine bonds, and the way that they could do that relied on the fact that cytosine and guanine form 3 hydrogen bonds opposite one another in a double-stranded DNA molecule, whereas thymine and adenine nucleotides only form 2, which means that cytosine-guanine pairs are more thermally stable than adenine-thymine pairs. So they would take strands of DNA and they would put them in a machine and shine light on them, and as they were increasing the temperature, they would watch the absorbance of those pieces of DNA over time, and the absorbance would remain the same, and then all of a sudden the absorbance would go up really high, and that was determined to be the melting point of the molecule of DNA. So the higher the melting point, the higher the GC content of that bacterium. So what that allowed them to do was not determine exactly what the GC content was, but it allowed them to determine and separate bacteria into high GC content bacteria and low GC content bacteria. And the reason I wanted to mention that today is because when you get to Berge's manual, you're going to see 2 of the major distinctions there are groups of bacteria known as high GC and low GC. And again, when we look at the prokaryotes, we see high GC gram-positive bacteria and low GC gram-positive bacteria. So I just wanted to give you a little bit of introduction to that before you were exposed to that verbiage in Berge's manual later on in lab. The other thing that I wanted to talk to you about today, just as it relates to the idea of looking at characteristics in bacteria in order to identify unknowns, is that we have a ton of special media types available to us in this class that you're going to be getting familiar with in the minor unknown and then utilizing in the staph, strep, enteric, and major unknowns to identify microorganisms. So I thought it might be fun to look at a couple of them here. The first one that we're going to look at of these 2 differential and selective media is mannitol salt agar, shown here on the left. So mannitol salt agar contains mannitol and salt. The mannitol, which is a carbohydrate, is something that some organisms are capable of fermenting and some are not. So this medium will differentiate mannitol fermenters from non-fermenters of mannitol, much the same way that the gram-stain reaction will differentiate gram-positive from gram-negative. And the way that this medium can do that is because in addition to mannitol and salt, it also has a pH indicator called phenol red, which, as its name suggests, is red at some pH, but it actually turns yellow when the pH decreases. So when an acid is formed, the phenol red will turn from red to yellow. So when you put organisms onto mannitol salt agar, if they are capable of fermenting the mannitol to acid, they will release that acid. The acid will lower the pH of the medium, and the medium will turn yellow. And that is a differential test for members of genus Staphylococcus. Staphylococcus aureus is able to ferment mannitol, whereas Staphylococcus epidermidis is not. So both of them grow, but only Staph aureus ferments mannitol, so you can differentiate mannitol fermentation on this medium, which is a really nice test for determining what your unknown staph is, and that's one of the tests that you're going to be looking at later on in lab. The other thing that this medium has is a selective ingredient. Now, selective ingredients, unlike differential ingredients, are not meant to differentiate the organisms. They are meant to select for organisms who have a particular trait. So when you have salt in the medium, only those organisms that can tolerate salt will grow, and so this medium selects for salt-tolerant or halotolerant organisms. So when we look at this plate, we can tell 2 things about these organisms. We can tell that both Staphylococcus epidermidis and Staphylococcus aureus are salt-tolerant or halotolerant. We can also tell that Staph epidermidis is not a mannitol fermenter, but Staph aureus is. So this medium is both differential and selective. It will allow you to look at the difference between two organisms, and it will also select for only some organisms to grow. On the other side of the slide, we have a different kind of differential and selective medium. This is called eosine methylene blue agar, or EMB, and EMB has two things in it. One is lactose and the other is bile salts. It also contains two dyes, eosin and methylene blue. So whereas the MSA contained phenol red to indicate a change in pH, this EMB contains eosin and methylene blue to indicate a change in pH. And that pH here does not happen as a result of mannitol fermentation. The pH change in EMB happens as a result of lactose fermentation. So Escherichia coli, which becomes highly pigmented because it takes up those dyes, is a lactose fermenter, whereas Pseudomonas aeruginosa, who does not take up the dyes and does not become brightly pigmented, does not ferment lactose. And just like the comparison between Staphylococcus epidermidis and Staph aureus allows us to see a potential pathogen in Staph aureus, the ability to ferment lactose will also differentiate normal flora from pathogens. In this case, though, the E. coli, which does ferment lactose, is part of the normal flora, whereas Pseudomonas aeruginosa, which cannot ferment lactose, is a pathogen. So again, this medium will allow you to differentiate between organisms based on their ability to ferment lactose. And that, again, is something that you're going to be looking at later on because you're going to have an enteric unknown. And one of the things that you need to know about that enteric unknown is, do you ferment lactose or not? Now, this is also considered to be a selective medium, whereas MSA had salt as a selective ingredient. EMB has bile salts as a selective ingredient, which may sound familiar, right? Bile salts are what you produce in your body and you excrete into your intestine in order to digest fats. And it makes sense that those organisms that inhabit the gut would be able to grow in the presence of the bile salt. So it turns out that bile salts are selective for gram-negatives and against gram-positives. So if you grow on EMB, chances are you are a gram-negative bacterium. So this selects for gram-negative bacteria, whereas MSA selects for halotolerant bacteria. So I know that's a lot of information about those two kinds of media. That's really more just to kind of prepare you for lab. For lecture exams and for lecture purposes, I think just understanding that there are such things as differential and selective media, and they're really useful because they tell us something about the physiology of organisms, and that using that information can be helpful in identifying unknown bacteria is really where you want to focus your attention. All right, before we go, just another look at how morphology and physiology can be used to identify unknown microorganisms. You can see here a key, and this will look familiar to you in a few weeks when you start looking at your major unknowns. We're differentiating bacteria based on their gram reaction. These are all gram-negative, and then on their morphology, we're separating bacilli from cocci. Then we're separating organisms based on how they use oxygen, whether or not they're motile, whether or not they ferment lactose, and that allows us to very quickly group our unknown bacterium into one of just a few genera once we know they're gram-negative. So morphology and physiology is really important in the identification of unknown bacteria. And because relatively few tests can be used to great clinical advantage, some of these tests that identify unknown bacteria have been grouped together. For instance, in trying to determine what kind of enteric bacteria you have in a patient specimen, you might use what's called the IMVIC test. And IMVIC is an acronym that stands for indole, methyl red, Voges proscure, and citrate. And what that means is You look at 4 physiological tests: whether an organism can produce indole from the amino acid tryptophan, whether it ferments glucose to mixed acids in the methyl red test, whether it ferments glucose to butadiol in the Voges-Presque test, and whether or not it's able to utilize citrate on this Simmons citrate agar. And you use those 4 results to give you a very quick shot at ruling out lots of organisms and keeping just a few for identification. So in this example here, and I know this won't mean anything to you yet, it will, don't worry, you have a positive indole result, a positive methyl red result, and negative results for butadiol fermentation and citrate. So you get a plus plus, negative negative, or minus minus code. So that means that the only possibilities for this organism are Escherichia coli, Edwardsia latarda, or Proteus vulgaris. And the cool thing about these organisms is that the ability to ferment lactose will quickly either rule in or rule out Escherichia coli, and the ability to swarm will quickly rule in or rule out Proteus vulgaris. So again, just a few tests and you're able to make an identification of your gram-negative bacillus. So, like I said in the previous slide, it doesn't mean you need to know all of this or memorize this chart for a lecture exam. This is just to give you an idea of how morphology and physiology are used to identify unknown microbes, right? So the take home is genetic analysis is used for classification and phylogeny, and morphology and physiology is most often used for identification, right? Giving a name to an unknown bacterium.